Hello, and welcome to our pre-lab activity associated with the cartilage lecture and the cartilage lab. Now, one of the things to keep in mind as we're looking at cartilage is we're looking at an example of a specialized connective tissue, but it's still a connective tissue. And so it's going to have the same characteristics as the connective tissues we looked at in the previous lecture, in the previous laboratory activity. So we're going to have cells that are going to be scattered away from one another. In this case, the cells are going to be cartilage cells, they're going to be chondrocytes. And in between them, they're going to have that extracellular matrix. And as a specialized connective tissue, skeletal connective tissue in cartilage, that extracellular matrix is going to be the predominant structure, and most importantly, it's going to be the ground substance that we're going to be looking at. Keep in mind that when we're looking at cartilage, cartilage is going to be avascular. There aren't going to be blood vessels that are going to be going into cartilage, and so it's going to be important that the cartilage should be able to allow diffusion to occur, because that diffusion is to bring oxygen and nutrients into the cartilage, bring it to the cartilage cells, as well as allow diffusion of the waste materials away from these cells. If we take a look at the extracellular matrix, the components are going to be type 2 collagen. That's going to be the characteristic matrix component. So type 2, again, a finer type of collagen than the type 1 that we looked at previously. And so we're going to have kind of a smoother, almost a glassy appearance to the connective tissue within cartilage. Lots and lots of ground substance, again, giving it a firm gel-like consistently, lots of aminoglycans or proteoglycans, drawing water in so that you can take a cartilage, you can compress it, squeeze the water out, remove the stress, and it's like a sponge. It's going to draw the water back in and essentially reinflate itself almost. If we take a look at it, again, focus in on this as a specialized connective tissue. And so we're going to have cells that are going to be scattered away from one another. Within the cartilage, the cells are going to be known as chondrocytes. And so what we're going to see are kind of rounder or oval cells. They're often going to have an eccentric nucleus. Keep in mind that these cells are going to be involved with synthesizing and maintaining the extracellular matrix. So they're going to have a prominent nucleolus, basophilic cytoplasm, because they're going to need to have a well-developed rough endoplasmic reticulum and Golgi apparatus. They need to be able to synthesize the proteins, modify and package the proteins for secretion into that extracellular space. Now, keep in mind again that the cartilage as a tissue is going to be avascular. We're not going to have a blood supply coming in. So these chondrocytes are going to be using anaerobic glycolysis because they're not going to be getting a huge oxygen supply getting to them. So they need to be able to do glycolysis to produce the ATP they need to do the things they need to do to maintain the matrix, but they're not going to be as efficient at it. If we take a look at the outside area of the cartilage, again in this one, we can see the cartilage in here to this point. Lots of really good cartilage cells here, kind of a nice even staining appearance throughout the rest of the cartilage matrix. Out here, we're going to have the perichondrium. Peri for around, chondrium for chondro for cartilage. We got the area around the cartilage. Keep in mind that this is going to be kind of a connective tissue layer that's going to anchor the cartilage to the surrounding tissues. And if we look at this, the perichondrium area, in great detail, a little bit higher magnification, we're going to see that there's actually two layers. We've got this outer fibrous layer, and this is going to be fibroblasts and collagen fibers like we've seen in the connective tissue proper. And they're going to be involved, in essence, with anchoring the cartilage to the surrounding tissues. And so kind of a connective tissue like generic connective tissue or connective tissue proper like function. Now, deeper to this fibrous layer, deeper to the fibrous layer over here, are going to be the chondrogenic layer. Chondro for cartilage, genic for birth. This is going to be an inner layer of, in essence, chondrocyte stem cells, relatively undifferentiated cells that, if they're activated, can start to produce cartilage. So they differentiate and they essentially add cartilage onto this outer surface here, pushing the perichondrium out, but increasing the mass of the cartilage. Now, if we look at the cartilage tissue itself, again, what we're going to be looking at, chondrocytes here kind of scattered around, extracellular material in between them, so type 2 collagen, abundant ground substance between it, giving a relatively smooth staining appearance. We're going to be looking at the fact that these cartilage cells, these chondrocytes, are going to be sitting within a space within the cartilage. Normally they're going to completely inflate and fill up the space. Sometimes when we're processing the tissues, they'll separate from the wall structure a little bit and may look like they're open, but under normal biological living situations, the cells are going to be pressed up against the surrounding connective tissue matrix, cartilage matrix. Now, so they're going to be sitting within a lacuna. Now, occasionally what you're going to see is that these cells are going to be dividing, but still trapped within a single lacuna. And these are going to be referred to as isogeny groups. We've got an isogeny group here. We've got another one over here. So we've got the products of a meiosis. As these cells are dividing, they're essentially adding to the cartilage mass. They're going to eventually probably produce some cartilage in between them and push themselves apart. So this is going to be that interstitial growth, that growth from within that we talked about is possible with cartilages. Again, because of the characteristics of the cartilage matrix, they're able to be flexible and essentially expand. Now, if we look at the cartilage matrix itself, again, kind of a smooth, glassy appearance, but we can see that there's difference in staining appearance. We can see a darker basophilic region kind of immediately around the chondrocytes and the chondrocytes within the isogeny groups. And then a little bit further away, we got a less basophilic staining appearance, a more kind of pinkish staining appearance. Now, overall, we're going to have this kind of bluish, pinkish staining appearance, giving it often kind of a grayish or kind of light purplish appearance, which is going to be characteristic of cartilage, especially hyaline cartilage. Now, the intensely basophilic region immediately surrounding the lacunae are going to be referred to as the capsular matrix. And it's going to be very basophilic because we have a higher concentration of the sulfated glycosaminoglycans and a lower concentration of the collagens. So we got the collagens all the way through this matrix, type 2 collagen, again characteristic of hyaline cartilage. But in essence, these glycosaminoglycans are going to be sticky. They're going to be at a higher concentration as they're deposited by these chondrocytes, and less of it is going to be able to diffuse over here. So we got more of the bluish staining appearance, more of the basophilic staining appearance immediately around these chondrocytes within the capsular matrix. In the intercapsular matrix, this pinker staining region out here, we got less of the basophilic staining from the glycosaminoglycans and more of the collagen. And so again, more of the collagen. Collagen stains pink because of its protein characteristics, and so it's going to stain kind of a pinker region, or at least a less basophilic region in that intercapsular matrix area. So that was hyaline cartilage, the most common cartilage within the body. We also have elastic cartilage, and elastic cartilage is going to be very, very similar to hyaline cartilage with one major difference. Instead of just being that type 2 collagen fibers that we saw in hyaline cartilage, elastic cartilage is still going to have type 2, but it's also going to have a lot of elastic fibers. And so we're adding in elastic fibers. Again, remember the characteristics that they're like rubber bands. They're capable of stretching and twisting and expanding. So we're going to have a much more flexible or much more elastic tissue when we look at elastic cartilage as opposed to hyaline cartilage. So if you'd like to take a look at this, especially with a stain, which is going to allow you to see the elastic fibers, you can see instead of the nice smooth appearance that we had with hyaline cartilage, we can see the more fiber staining because we can see the elastic fibers in elastic cartilage. Again, chondrocytes, kind of the round cells, oval nuclei, eccentric nuclei, often, especially within elastic cartilage, lots more of those isogeny groups that we talked about. So multiple chondrocytes within a single lacunae. Overall characteristic is it's still relatively the same as hyaline cartilage, except it has the addition of those elastic fibers being present there. So it's still, these cells are going to be involved with maintaining the matrix. It's going to be an avascular tissue. We're going to have a perichondrium around the outside over here, both above and below this mass of elastic cartilage through this region right through here. Elastic cartilage, again, if we take a look at it in a fresh specimen, it's going to have yellowish when it's essentially just dissected from the body. Because of the presence of these elastic fibers, it's going to be much more flexible than hyaline cartilage, but structurally, it's going to be essentially the same as hyaline cartilage with the addition of that dense network of elastic fibers. And that network of elastic fibers then is going to give it that flexible characteristic. Again, we've got little rubber bands in here, in essence, providing us with some structural support within this tissue. And then the final type of cartilage we're going to look at, relatively rare type of cartilage, is going to be the fibrocartilage. And now with the fibrocartilage, we're going to break some of the rules that we have with hyaline cartilage and elastic cartilage. Hyaline cartilage and elastic cartilage had type 1 collagen. With fibrocartilage, we're going to be back with type 1. Type 1, thick cartilage, thick collagen bundles packed in close to one another. And so it's going to be a little bit more similar to a connective tissue proper because we've got the type 1 collagen, but we still have a lot of those ground substance, a lot of the proteoglycans, the glycosaminoglycans that are going to be present there. And so it's got that extra added component, the ground substance, which helps make this into a cartilage as opposed to a more generic connective tissue proper. We take a look at the cells. Again, these cells are going to be rounder, they're going to be chondrocytes, cartilage cells, often in columnar isogenous groups. And so if what you're looking at is kind of a dense, almost regular connective tissue appearance, but instead of those flat nuclei that we saw with fibroblasts, we see round nuclei, round cells, and they're kind of stacked up in little rows. We should think about this as being fibrocartilage. So it resembles dense connective tissue, but the cells are going to be different. Instead of the flat fibroblasts, we're going to see these round chondrocytes. If we take a look at the actual molecules that are present, in addition to the type 1 collagen, which we would have in a dense connective tissue, we're also going to have lots of ground substance in there as well. And this finishes up our overview of the cartilage lab slides and cartilage concepts. Hopefully you'll be able to use this information, be able to look at the slides, look at the images, and be able to identify the structures that we're looking at. As always, if you have any questions, feel free to email me at hoffmanj@arcadia.edu. Thank you.